Okay, so hi everyone. Today we are going to be looking at what we call synapses, okay? So basically we have got two types of synapse, and we have got what we call electrical type of synapse, and we also have got what we call the chemical type of synapse. So now today I'm going to focus much more on the chemical type of synapse because the electrical type of synapse is not that complex, and it's found in what we call cardiac cells, okay? So in cardiac cells, the type of synapse that is found is known as electrical type of synapse. So now if you look at the nature of cardiac cells, we have got what we call intercalated discs. And in between the intercalated discs, there's what we call gap junctions. So those gap junctions form what we call synapses. So it's just basically transmission of electrical signals from one cell to the other, which forms what we call a essential. So that essential allows the transmission of electrical impulses at a very fast rate, and it does not involve, it does not take time. It's much faster and more efficient. You want to find that the arterial cells contract at once, and also the ventricle cells contract at once. Also look at the hearts, the human heart physiology in terms of contraction. You're going to find that it's myogenic, meaning that the electrical signals or the impulses arises from the heart itself via what we call the pacemaker cells. So now today we are going to be looking at what we call the neuromuscular junction, which is an example of what we call as a chemical type of synapse. So a chemical type of synapse is just between cell-to-cell communication. So what you're seeing there is a neuromuscular junction. If you look at the word neuromuscular junction, it's just a connection of two words. There's a neural part and there's a muscle. So it's a connection between a cell from the nervous system and also a muscle. Okay, so you're trying to pass on information from the nervous system to the muscle, and that junction or that communication pathway is known as the neuromuscular junction. That's what we are going to be looking at. I'm sure this video will be very short. It can even be less than 10 minutes or max maybe less than 15. Okay, yeah. So now the events occurring during transmission of impulses from the motor nerve to the muscle are somewhat similar to those occurring at neuron to neural synapses. So now what you're seeing on this diagram is just trying to show a neuromuscular junction. So as you fiber, right? So this motor nerve fiber is being seen, which has got what we call a myelin sheath. So now when you talk about the myelin sheath, it just has to do with increasing the speed, which is made up of a fat layer or a lipid layer, which acts as an insulator so that no charges are lost as the impulses are being transmitted. And it also helps the impulses. Lack of a better word, we say jumping, okay? Meaning they'll be moving from at a faster rate as compared to a non-myelinated type of nerve fiber, okay? So basically what you're seeing there is the axon terminal, as you can see on this image, which has got what we call mitochondria there, and has got vesicles containing ACH or acetylcholine. Then we also have got what we call the Schwann cells. Basically, the Schwann cells, as you know, they're the ones which are responsible for producing the myelin sheath. Okay, so the Schwann cells produce this lipid that goes around this motor nerve fiber. Then you can see it's showing active zones. So these are areas where the vesicles are able to undergo what we call exocytosis. Exocytosis basically simply means one form of bulk transport, okay? Yeah, amongst the different type of transport system. And bulk transport is known as an active process, meaning it requires energy. So you'll be seeing as we go on that there will be some active process happening known as exocytosis, okay? Meaning substance will be expelled to the outside. All right. Then there's a space in between these two adjacent cells. You can see between the muscle and the neuron, there's a space here, and this is known as the synaptic cleft. And as you go to the muscle, you can see this is known as the sarcolemma. Sarcolemma simply means the membrane, the cell membrane of a muscle cell. It has got a distinct name, which is known as the sarcolemma, as you can see. Then we have got this junction of folds, as you can be able to see, which are there. All right. Which will be more important because that's where charges will be flowing in when a muscle action potential is triggered. Then there's a nucleus of muscle fiber there. As you know, muscle fibers or skeletal muscle fibers are multinucleated, unlike cardiac or smooth muscle cells. So we'll be talking about what happens there. then region of sarcolemma with ACH receptors. So as you know, in order for an action potential to be triggered on the other side of the cell or the muscle, you're going to find that we need ACH receptors, which are known as nicotinic ACH receptors. All right. Yeah. So let's go to the next slide. So now the impulse arriving in the end of the motor neuron increases the permeability of its endings to caution. Okay. So I like to explain this with the aid of a diagram. So let's just go through the text, then we go to the diagram, we'll be able to explain better. Then caution enters the endings and triggers a marked increase in exocytosis of the acetylcholine containing vesicles. The acetylcholine diffuses to the muscle type nicotinic acetylcholine receptors. Then there is an increase in the sodium and potassium conductancy of the membrane, and the resultant efflux of the sodium produces a polarizing potential called the end plate potential. All right. So now acetylcholine is then removed from the synaptic cleft by acetylcholine hysteresis, which is an enzyme, which is present in high concentration at the neuromuscular junction. All right. So now let's look at this diagram. So this diagram has got about 8 stages that you are going to explain. When you are done with these 8 stages, that means we are done with the neuromuscular junction. You are even able to write an essay and get everything correct. So now here is a diagram showing an action potential coming via the motor nerve fiber. All right. So there's a motor neuron there. So you're going to realize that there's an action potential coming. So now, if you have learned cell physiology before and you have learned how an action potential is triggered and how it happens, this video will be very easy for you. For example, if you look at how we have got about 3 stages. There's a resting membrane potential, then an action potential. There's resting membrane potential, a graded potential, then action potential. So now, in order for us to move from resting membrane potential to an action potential, there has to be a graded potential. So this allows us to reach what we call a threshold potential. So once we attain a threshold potential, then it can trigger what we call an action potential. And an action potential involves what we call depolarization and repolarization. So depolarization has to do with the opening of the sodium variegated channels. Okay, so these are known as sodium variegated channels. When they open, they allow the influx. Okay, so there's going to be sodium coming from the outside, then going inside the cell. That is known as depolarization, meaning the inside part of the cell is going to become positive, because initially the way it is, the inside part of the cell is negative, then the outside is positive. So as sodium is going into the cell, which is a more electropositive electrolyte, it's going to change the charge for the inside to become more positive than the outside. That is known as depolarization. Then when you talk about repolarization, it has to do with the opening of what we call the potassium variegated channels. And remember, these channels are not open at the same time. When sodium closes, potassium opens, all right? So that's how an action potential is generated. So for repolarization, the potassium variegated channels are going to move from the outside, from the inside, and go outside, because as you know, there's more potassium inside the cell and there's more sodium outside the cell. So as they are moving via what we call facilitated diffusion, hence it's called facilitated diffusion, because we require what we call a channel protein, meaning you require something in order for that to happen. So that's basically what it means when you talk about an action potential. Then there's also what we call hyperpolarization, okay? There's what we call hyperpolarization. This has to do with the delay in the closure of what we call the potassium variegated channels. But once, together with the leaky channels and the sodium potassium variegated channels, together with the sodium potassium pump, it can now go back to its resting membrane potential. So basically that explains what an action potential is, but I'll send a video which will talk about that in detail later. All right, then when you look at this, so now as the action potential is coming in, you're going to realize that the moment it reaches at the motor end plate, where we have the mitochondria and vesicles, there's going to be what we call the calcium variegated channels are going to open due to the charge that has arrived at this particular area. So once the calcium enters into the cell, it's going to cause, okay, so once the calcium enters the cell, it's going to cause the vesicles to migrate towards the active zone. And when they reach the active zone, there's a phenomenon that happens there, which is called exocytosis, meaning there will be the emptying of the vesicles to the synaptic cleft, okay? But as this is a form of active transport, energy is going to be received from, or it's going to be supplied by the mitochondria via, in form of what we call ATP. So now, at this stage, there'll be exocytosis. Under exocytosis at the active zone, these vesicles will rupture to release the ACH in the synaptic cleft, okay? So this is stage 3, which has to do with acetylcholine release, as you can see. So now, as acetylcholine is being released there, so like in the previous slide, you saw that here we have what we call receptors, okay? And these receptors are known as acetylcholine nicotinic receptors. So these are able to catch, okay, or the acetyl is going to, when it lands on the these receptors, they are going to cause the opening, okay, of what we call the sodium vorticity channels, which are there, or the channels on the other side of the membrane, which is going to cause the generation of what we call a muscle action potential, all right? Then, if that action potential is generated at stage is generated, it's going to allow the entry of sodium, or in short, it's going, the ACH are going to cause the opening of the sodium channels and allow the entry of sodium into the cell. So as sodium is entering into the cell, it's going to cause the same thing or same phenomenon, which we call depolarization. Then that depolarization is going to trigger a muscle action potential starting. Then when you go to 5, it's saying local current between depolarized and end plate, local current between depolarized end plate and adjacent muscle plasma membrane. So there's going to be another muscle action potential which is going to be generated on the other side. Yeah, and we talked about how that happens. Then at 6, muscle fiber action potential initiation, right? Then at 7, we are going to, there will be propagation of action potential in muscle plasma membrane. So that propagation of muscle action potential in the plasma membrane is the one that is going to go down in those T-tubules, hence causing what we call opening of what we call the sarcoplasmic reticulum by using the rhyodine receptors, etc., just like that. Yeah. Then at stage 8, which is the last stage, which has to do with the breaking down or degradation of the ACH on the receptors, okay, using an enzyme known as acetylcholine hysteresis, okay, on the motor end plate. So because when the acetylcholine remains on this, it will keep on showing that there is supposed to be an action, there's already an action potential happening. So it has to be broken down, then allow the muscles to relax, then again an action potential is triggered, etc. So I think this is the end of a video on this type of synapse, okay? So apart from this, there's also what we call chemical, neuron to neuron, but this example is known as neuron to muscle, and it's the one they most bring during exams. And thank you very much for watching this video, and have a blessed day.